Welcome. In this session, we'll address the visual pathways, both the anatomy and the effect of lesions to the visual pathway. We will identify the anatomical components of the globe itself and the functional elements of that globe. We'll identify the various diseases that affect the globe, and that includes cataract due to refractive error, and opacity of the lens. And then we'll look at glaucoma due to increased intraocular pressure. We'll look at retinal diseases that include vascular diseases such as stroke, diabetes, and macular degeneration. We'll look at optic nerve degeneration associated with glaucoma and optic neuritis. And then photoreceptor and retinal pigment epithelial disorders such as color blindness and retinitis pigmentosa. We'll define the terms used to describe the optical properties of the eye. We'll identify the major structures in the visual pathways, starting out in the retina, going into the optic nerves, the optic chiasm, the optic tract, the lateral geniculate, optic radiations, striate cortex, which include the lingual gyrus, calcarine fissure, and the cuneus. We'll draw the course of the nasal and temporal retinal fibers from each eye to the visual pathway, because they get divided up at the chiasm and go to separate hemispheres. We'll describe the visual field losses produced by lesions at various locations along the visual pathway and identify the terminology of each type of visual field deficit. We'll identify the pathways and structures involved with a couple of specialized reflexes and functions. The light reflex for miosis, the narrowing of the pupil, the accommodation reflex for objects that are brought close to the body, to the eye, and we'll look at pupillary dilation, that is mydriasis, opening up of the pupil. And then we'll identify the neuroanatomical substrates for the Argyll-Robertson pupil, Adie's pupil, and the Horner syndrome. We see here an artist drawing of the entire visual system, starting from the globe and the retina, through the optic nerve, chiasm, optic tract, lateral geniculate in the thalamus, and radiations back to the cortex. And those are all outlined here. We see the retina, optic nerve, the chiasm, the optic tract, the synapse in the thalamus, which is the first synapse after leaving the retina, in the lateral geniculate nucleus of the thalamus, and then the thalamocortical radiations to the visual cortex, both on midline and mid-occipital, midline in the occipital cortex, and on the external surface of the visual cortex. If we look at the eye itself, we can see three different layers. We'll see the cornea, and here a clear structure, and the sclera, that is a very dense connective tissue that is non-distensible and essentially maintains the outside structure of the eye. It is traversed by vessels that feed the retina, as well as by the nerves that travel information to the rest of the brain. We can see then the middle coat of the three, and that is the uvea. And the middle coat is pigmented, and it includes the iris that we see right here, and the ciliary body that we see right here, as well as the choroid, this blue shell that is highly vascularized and extends as far as the retina extends. And then finally, internally, we'll see the retina, which is this green shell, and the pigment epithelium, which is immediately behind it. The inner coat is the neurosensory component, the retina, and that's where the photoreceptors, the receptors that actually transduce light, sit. And that retina is part of the optic cup, optic vesicle and cup, and is in fact part of the central nervous system. The only part of the central nervous system that's visible from the outside, literally by looking through an ophthalmoscope. And the optic cup often also gives rise to the retinal pigment epithelium. We see here a cross-section through the retina with the vitreous in this most superior position, if we had cut through the eye. And then we see the nerve fiber layers. These are the fibers that are going to go back to the optic nerve. We see the ganglion cell layer, which are the cell bodies for the nerve fiber. We see the inner plexiform layer, the internuclear layer, made up of bipolar cells, who have cell bodies here, the outer plexiform layer, the outer nuclear layer, and these are the cell bodies of the photoreceptors, and we see the photoreceptors right here. And just adjacent to the photoreceptors, we see the retinal pigment epithelium. And just outside the retinal pigment epithelium, we see the choroid, which is the vascular, highly vascular tissue to supply the outer portion of the retina. And then outside the choroid, we see the sclera, which provides the firm support for the excuse me. Here we see the view through an ophthalmoscope of the retina. We see two major structures, and that is the macula, the central portion of the retina, which is relatively avascular and has the high concentration of cones responsible for your very best vision and for your color vision. And we also see the optic disc, and the optic disc is where all of the axons coming away from the photoreceptors that had fed into the ganglion cells. The ganglion cell axons all end up coming into this optic disc and then begin to form the optic nerve as they head out. So this is the optic nerve head, the optic disc. And we see all the retinal vessels that come in through this optic disc. If we do a cross-section through this retina, then we can see the fovea, this region right where the macula is in the very center of it, has essentially no ganglion cells and little or no of these inner plexiform layers, but has a high concentration of photoreceptors, particularly of the cones that are responsible for vision. Visual acuity is dependent to a significant extent by the properties of both the pupil, that is the cornea, and the lens, and in particular the lens relative to the length of the eye. And in a normal individual, images of structures outside the eye are focused directly on the retina. In a myope, a person who is nearsighted, the image is focused inside the globe, and lenses are needed to correct that vision in order to have the image located on the retina. In farsighted individuals, the image would end up being focused behind the eye, and lenses are necessary to bring that image closer to rest on the retina. Normal vision is emmetropic. We can see some portions of the visual pathway from the base of the brain. The globe has been removed, but we see here the optic nerve, we see the optic chiasm, and the optic tract. And we see the tracts going in each direction. We see here, of course, the midbrain. And then we also see, in this location, the mammillary bodies. And here we see the superior colliculi. But we can also see, as we go further back, the lingual gyrus near the midline that gets visual input, the occipital pole, the occipital gyrus, and the occipital temporal gyrus. And we can see some aspect of those in this MRI image. And in particular, we can see that in this region is where the optic tract would come through. And we can see the optic tract a little bit over here. Then occipital cortex would be right back here, occipital cortex right here. Now, the visual fields are of particular interest because they give us a very good indication of the way in which the world is represented by the two eyes and how the images of the two eyes come together within the brain. Each eye is able to see a large portion of the world, about 180 degrees, and we divide that world in half and talk about an ipsilateral and contralateral portion that will follow up, and ipsilateral and contralateral hemifields. When we look at the world with the two eyes, the input from the two eyes overlaps substantially, so they see pretty much the same world, but there is a monocular crescent, so each eye sees a little bit on its side of the world that the other eye doesn't see. But we're able to fuse an image in this central area and see a unified image. We refer to the area on each side as a hemifield. If we split the world in a vertical with this vertical line, we refer to this as a hemifield, and we refer to each of these quarters as a quadrant. And we recognize that the fovea is in the very middle for each eye of that visual field, and that there's a blind spot. We see it represented here by a little black spot, dark spot in each eye, and that is the location of the optic disc. It's where the optic nerve leaves the globe, and there are no photoreceptors at the optic disc. Now, we'd like to follow the pathway, in this case, from a given eye, and in this case we're going to look at the right eye, and as we look at the right eye, we see that as it looks out on the world, the left visual hemifield comes into the eye, and because of the lens, it's picked up by the right side of the retina of the right eye. Those fibers will travel through the optic nerve, through the chiasm, and they will then stay on the same side. They will stay on the same side of the body and travel back through the optic tract to the lateral geniculate. From the lateral geniculate, they either go straight back to the visual cortex, or they go through the temporal lobe and then back to the visual cortex. If we look at the input from the other eye, from the left eye, it's also seeing this left visual field. It will come into this hemiretina, travel through the optic tract, optic nerve, to the chiasm. Those from this right side, that is the nasal side of the left eye, will travel across the chiasm to the opposite side to join with its partner that represents the left visual hemifield. They'll come together and they all synapse at the lateral geniculate nucleus and then travel back to the visual cortex. As a result, the visual cortex is getting information from each eye from the opposite hemifield. So the right visual cortex gets input from the left visual hemifield of the left eye and of the right eye. That has important consequences for the nature of deficits that subsequently occur if there is damage anywhere along this visual path. And we see that represented here. If we have damage to either the retina in one eye or to the optic nerve, particularly if we cut the optic nerve, then you would lose vision in the optic nerve, in the right eye, in both fields, in the left and the right hemifield, but have no effect on the left eye. So this would be the visual field defect, a blind right eye, if you were to cut the optic nerve of the right eye. If you were to damage the crossing fibers at the chiasm, the optic chiasm, then the crossing fibers are not able to reach the cortex. So those coming from this hemiretina will be able to travel back and stay in the same side. Those traveling from this hemiretina will be able to travel on the same side and stay on the same side. The resulting vision will be that person would have a bitemporal hemianopia. They would be able to see in the nasal field, that is in the right visual field of the left eye, and they'd be able to see in the left hemifield of the right eye. That's a bitemporal hemianopia, and it happens under conditions in which this area is damaged, the chiasm itself, right at the midline, and right under the chiasm is the pituitary gland. So pituitary tumors, as they grow up, have a tendency to start to produce this effect. They can produce that effect, or they can produce a partial effect of a bitemporal hemianopia. If there is damage just at this level of the chiasm, but just at the lateral portion of the chiasm, then it would block the fibers coming from this temporal hemiretina, as we can see here. That temporal hemiretina is seeing the left hemifield of the right eye. This person would have a left hemianopia of the right eye, only of the right eye, and have normal vision in the left eye. If there is damage that cuts through the optic tract, right here on the right, then all of the fibers coming to the right visual cortex would be lost with blockage here, the optic tract. That would produce a left homonymous hemianopia, where the person would be able to see in the right visual field of each eye, but have no vision in the left visual field of each eye, because these pathways have been lost. As we go back to the geniculate, we see that the fibers will start to separate out, and those that come from, some go into the temporal lobe in an area that's called Meyer's loop, and then travel back to the visual cortex. If there's damage in the temporal lobe to these fibers, those are fibers that represent the superior portion of the opposite field, that is, this is on the right side, of the left visual field. So this person with damage to Meyer's loop would have a left superior quadrantanopia, a loss of vision called a pie in the sky. On the left would tell you that there's damage in the right hemisphere. If there's damage to both the pathways that are going directly to the cortex from the geniculate and through the Meyer's loop, if all of these fibers are cut, then you'd have a left homonymous hemianopia, and it might look just like this damage when you have the optic tract cut. It's just that the optic tract is a very narrow structure, so it's easy to hit the entire optic tract. The optic radiations are spread out a little bit more, so it's less common that at this location you would hit all of the fibers, but if you do, you would have a left homonymous hemianopsia. As you come back to the cortex, the separation between the upper and lower portions of the hemifield are sustained at the primary visual cortex. And the calcarein sulcus in the visual cortex separates out upper and lower portions of the visual field, of that visual hemifield. As a result, if you had damage to just below the lower portion of the calcarine fissure, this lower bank represents the upper portion of the visual world, and you'd have a left superior quadrantenopia, similar to what you have over here. And if you had the opposite region, that is the upper bank of the calcarine cortex, then you would have a left inferior quadrantenopia. If you had substantial damage to the visual cortex that damaged the upper and lower banks of the calcarine cortex, you would have a left homonymous hemianopsia, just as you have here at the optic radiations and just as you have at the tract. So it is possible to have a homonymous hemianopsia, damage to these two areas, damage here, or damage here. So they might be a little bit ambiguous, but when you have damage to just the optic nerve or to the chiasm at this location or to Meyer's loop, you have these rather distinctive features in which there is a very different pattern from those when you see these homonymous deficits for full field loss at this location and at this location, and a combination of these two locations. Also, the visual system has multiple subcortical control systems that optimize vision. One is pupil, and that is pupillary constriction, optimizes the amount of light coming to the eye to get the best visual screen, or to just best visual image and reduce scatter, but also it is a way of testing the visual pathway. And it controls both pupillary constriction and pupillary dilatation. And then we'll look at accommodation in which there is a lens thickening, pupillary constriction, and convergence of the eyes to see some objects that are closer, and a variety of eye movements controlled by the cranial nerves 3, 4, and 7. We'll look first at pupillary control and the pupillary light reflex. Stimulation of one eye, light comes into the eye, activates the photoreceptors and the optic nerve. The optic nerve sends its inputs. In addition to the pathways that we talked about earlier that go to the lateral geniculate, there are pathways that go over to the pretectal area. So we see the pathway that went to the lateral geniculate, but there are also fibers that go to the pretectal area. And from the pretectal area, we see this is the midbrain. Fibers travel to the Edinger-Westphal nucleus, a portion of the third nerve nucleus. And from there, out in cranial nerve 3, out to the pupil to induce closure of the pupil. That's the direct light reflex. It's pupillary light reflex, and it's dependent on optic nerve, cranial nerve 2, and cranial nerve 3. So it's a very good measure of the integrity of that pathway from cranial nerve 2 to cranial nerve 3, and involving the midbrain. Also involving, of course, the optic nerve, the chiasm, and the optic tract. Now, when you flash a light in one eye, it turns out that both eyes the pupil will close. That occurs because the input that we've just described that controls this direct light reflex also sends information across the posterior commissure, just above the periaqueductal gray, where we see the cerebral aqueduct, we see periaqueductal gray. The posterior commissure travels from one side to the other in the pretectal area and sends its fibers down to the Edinger-Westphal nucleus on the opposite side to activate the third nerve and cause pupillary constriction on the opposite side. That's the indirect or consensual reflex of the contralateral eye. Damage to the optic nerve on one side would affect pupillary response in both eyes, would be diminished, while damage to the posterior commissure would result in a normal direct reflex, but a blocked consensual reflex. And we'll see some elements of that. So we see here that if there was damage to the optic nerve in this eye, then if you shine a light in that eye, you would not have a pupillary response because there's no light getting and no information getting back to the Edenja-Westphal nucleus. But if you shine a light in the opposite eye, you'd get contraction of this eye because this pathway is intact, and you'd get contraction of the contralateral eye because the pathway through the commissure is intact and the third nerve is intact, so you can get pupillary constriction of the opposite eye. That separates out third nerve damage from from optic nerve damage and tells you have damage to the third nerve or to the Edenja-Westphal nucleus in that region. A tonic 80s pupil is a unit which we see here. This is a tonic pupil on this eye, is a unilateral mydriasis, pupillary dilatation, in which the pupil is unresponsive to direct or consensual stimulation. And it tells us that there's a peripheral lesion in the postganglionic sympathetic outflow from the Edenja-Westphal nucleus, cranial nerve 3. So if we have damage to cranial nerve 3, this pupil will be permanently dilated and will not respond to light. If you have damage to cranial nerve 3, even though light in the optic nerve may come in from either side and activate these structures, if the cranial nerve 3 is damaged, the output will not occur, and the pupil will stay open. Accommodation is fixation on a near object in which there is contraction of the ciliary muscles with lens thickening. There's pupillary meiosis and convergence of the two eyes, that is the medial rectus of each eye, is activated. This pathway depends on information coming to the cortex that is able to detect that the information is out of focus, and as you bring something closer to the face. So it starts out in the visual cortex, goes to the premotor frontal cortex, and from the frontal cortex it comes down through the internal capsule, through the corticobulbar fibers, to the oculomotor region of the third cranial nerve and the motor neurons of the subnucleus of the third cranial nerve that control activation of the medial rectus. And the preganglionic sympathetic neurons of the Edenja-Westphal nucleus activate contraction of the pupil, the parasympathetic neurons, and the ciliary muscles in order to get a change in the lens. In the Argyll-Robertson pupil, it's usually a bilateral defect. Both pupils do not react to light. That is, they do not have a direct light response, but they do constrict during accommodation. That is, accommodation is retained, but light is lost. What that means, it is usually caused by damage to the pretectal area, just up here at superior portion of the midbrain. And in this setting, it interrupts the light reflex to the Edenja-Westphal nucleus, but does not disturb the input that's coming from the cortex to the accommodation that comes through the crus cerebri, that comes down here, through the crus cerebri and up to the Edinge-Westphal nucleus. It is a distinct manifestation of one of the neural manifestations of syphilis. And finally, pupillary dilatation is controlled by sympathetic pathways from the hypothalamus. And they travel down through the lateral portions of the brainstem. Horner's syndrome is one manifestation of those, and that is meiosis, where you have pupillary constriction. It also includes ptosis, that is a lid drooping, and the absence of sweating on the same side, which is all ipsilateral to lesions of the sympathetic pathway in its descent through the brainstem, or its ascent from through the superior cervical ganglion. Thank you very much. If you have any questions, don't hesitate to give me a call.